So this is the second part of our identification of bacteria lecture, and where the last video left off was just finishing with what a differential versus a simple stain were. And so now we're going to be talking about three different examples of differential stains. So just a quick review, a differential stain is one in which there are two different dyes that are used. And so at the end, any bacteria is going to stain one of those two colors. And so that result then is going to give you additional information above and beyond what you'd obtain from a simple stain. So for example, the gram stain is going to determine which type of bacteria have either gram positive or gram negative. And those are going to be based upon the end result colors. So gram positive will stain purple during a gram stain, whereas gram negative at the end will stain pink. And so over the next couple of slides, what we want to look at is how a gram stain is actually done to achieve those results. One other thing before we do that is to realize that this difference that you're obtaining, gram positive versus gram negative, that difference is based upon the structure of the bacterial cell. So we haven't fully explained the structure of bacteria. That's going to be the next lecture when we look at prokaryotic cell structure. But for now, we can say that there's a structure on the outside of the bacterium called their cell envelope, and that is going to differ between gram positive and gram negative, and this stain will be able to pick up on that difference. So what does this stain actually look like? So there are going to be four different chemicals that are added to the bacteria during a gram stain. And the first thing that you need to do is get your bacteria that you're interested in onto a slide. So it's called making a bacterial smear. And then the first dye that's added, remember there are two dyes during a differential stain, which a gram stain reaction is a differential stain. So the first of two is called the primary stain. And so this primary stain is called crystal violet. And as the name indicates, it's going to be a purple color. And as with many of the dyes that we talked about, they are going to be positively charged. And so that's going to interact with the negatively charged bacterium. So that dye is going to sit on there, as it says, for one minute in length. And at the end of that step, you rinse off the dye with water, and both of the bacterial structure types, both of the cell envelope types, will stain the same color. They'll both be purple at this point in time. The next reagent that's added is an iodine solution. So what the iodine does, it's called a mordant. And the way I like to remember this, it almost sounds like the mortar you put in a wall. So it helps hold things in place. And so that's what a mordant is doing. It is going to form a reaction with the crystal violet to form a complex, so a larger molecular complex that's harder to remove from the bacterial cell envelope. So that's what it's doing. So it's not changing the color, but it's going to affect how readily though that complex is going to be removed from the cell envelope. So that's what the iodine is doing. So that's the second step. The third step is the addition of what's called the decolorizer. And so that tells you what its function is going to be. So it's made up of, sometimes it's just alcohol. In our lab, when we actually have labs, it is a mixture of both acetone and alcohol. And what that's going to do is affect the two different types of bacterial cell envelopes differently. So again, we haven't fully explained the cell envelope, so I'm just going to give you a quick little lesson here. The gram-positive bacteria have a thick cell wall, so we call this cell wall, it's made up of a repeating block or a repeating building block called peptidoglycan, and so it has many layers, and so it's thick. And so when you add the decolorizer to a gram-positive bacteria, that purple color is not stripped as readily from this thick peptidoglycan cell wall, and so the purple color is retained. By contrast, gram-negative bacteria do not have a thick cell wall. Instead, they have a thin layer of peptidoglycan, a thin layer of a cell wall. And so when this decolorizer is added, it's going to disrupt an outer membrane that exists only in gram-negative. So that gets disrupted. And then that decolorizer has access to that thin peptidoglycan cell wall, and the purple complex is not held as tightly in that thin layer, and so it can be stripped more rapidly. And so if the length of the application of decolorizer is correct, then what you get is the removal or the decolorization of gram-negative bacteria, whereas the gram-positive bacteria will still retain that purple color. So at the end, this is where you get a difference in result through the decolorization step. And so after this step is the fourth and final step, and this is the application of the second dye. So this is called the secondary stain, or sometimes it's called the counter stain. And in this case, it's a reddish pink dye. It's called safranin. And so gram-positive bacteria, since they still have a dark purple color, this dye isn't going to change their look at all. But gram-negative, since they are colorless, will take up that safranin, that counter stain, and so they will appear pink. So at the end result, when you put a bacterium through a gram stain, you're going to get either a purple result, and that would mean that's a gram-positive bacteria, or you're going to get a pink result, that is a gram-negative bacteria. So that's one example of a differential stain, because you are able to visualize the shape of the bacteria, the arrangement, and also the size, but now you can also see what color is the end result, and so that will give you a gram stain result. Is it gram positive or gram negative? So that's a really important stain that we use in microbiology. These are pictures of 4 different bacteria that were put through the gram stain. And so this bacteria, you can see that's an individual bacterial cell that I'm pointing to right there, and so you can see its shape. It's round, and it is purple in color, whereas there's a single bacteria. You can see that's much more elongated. It almost looks like a dash or like a hyphen. That still is also purple in color. So we can say both of these bacteria are purple, gram-positive, but they have different shapes. When you look over here, that's a single bacterium. So again, a circle in color. This also looks more like a dash or hyphen. And so we call that a rod-shaped bacteria. But these are both pink, and so we conclude these are gram-negative bacteria. So that's the power of the gram stain and how those results would be obtained. The gram stain isn't the only differential stain. Another fairly important differential stain is called the acid-fast stain. And so this is used really to identify here we have this is a genus. So Mycobacteria is a genus of bacteria, and Nocardia is a separate. And so these two genera of bacteria do not stain well with a gram stain, because they have an additional molecule in their cell wall that's not present in all bacteria. And so if bacteria have this waxy compound called mycolic acid, it makes it really hard to stain, and it gives them some other properties as well. This genus Mycobacterium includes the bacterium that causes tuberculosis. So that's probably the most important bacteria that's acid-fast, because across the globe, it has a fairly high disease burden and infects a lot of individuals across the planet. And so being able to identify if someone does have Mycobacterium in there can be really important. There's one other disease that's listed here under Mycobacterium, which is it causes leprosy as well. But again, tuberculosis is by far the most abundant acid-fast bacteria that is affecting humans on the planet. So the goal of the acid-fast being a differential stain, still to recognize the size, the shape, and arrangement of bacteria. But now we want to ask the question, is this mycolic acid present or is it not? And so that's how we define acid-fast bacteria. They have mycolic acid present. And so those that don't have mycolic acid, we call them non-acid-fast bacteria. So this differential stain called the acid-fast stain was going to have 3 different chemicals that are added. 2 of them are dyes, and so we're going to start with the first dye, and it's called carbofluoxin. So you can see the name here. Fuchsin is very similar to the word fuchsia, and so that gives you an idea of what color it is. So it's kind of a pink, purple, fuchsia, magenta-y color. And one other thing that's important to note is that acid-fast bacteria are extremely difficult to stain. Because of that waxy molecule called mycolic acid present, they really resist most chemical treatments. And so we have to use a much more strong way to kind of force the dye into them. And so what we do is we not only have a longer period of exposure, and this is as compared to a gram stain, which was a minute for the primary stain. So this is 5 times longer. But then we're also applying some heat source. So this varies with different acid-fast procedures, but when we carry this out in our laboratory, we use a slide warmer or a hot plate. And so 5 minutes of allowing the primary stain to really get forced in there. So remember the difference between these. Acid-fast bacteria have that waxy mycolic acid, and non-acid-fast don't. So if we have the conditions sufficient to force the dye in, then these will indeed stain that fuchsia color. And non-acid-fast bacteria, since they don't have that waxy molecule, will take up the stain much more readily. So at the end of the first step, at the end of the primary stain of carbofuchsin, all the bacteria should stain that fuchsia color. The second step is the decolorizer, and this is different than the gram stain in that its chemical composition is different. So alcohol is common across both stains, but notice this is unique. Having acid present, and this is in the name of the stain, it's called an acid fast. And so the acid fast bacteria that have that waxy mycolic acid resist this acid decolorizer. Another way of thinking of it is we're trying to acid wash these bacteria. And so acid fast bacteria are acid resistant. They resist this acid washing. The term that's used, they are acid fast. That's where the name comes from. And so they resist the decolorizer step, whereas non-acid fast can't resist that acid washing. And so their color, their primary stain color, that fuchsia, is stripped or decolorized from it. So again, at the end of the decolorizer is where you have one retains the original primary stain, the acid fast keep this pink color, whereas non-acid fast would be decolorized. Then we apply that second dye. This is called the secondary or counter stain. And this dye in acid fast procedure is called methylene blue. So it tells you again the color. The acid fasts that have the fuchsia color already embedded in it do not change their color. They keep that. Whereas those non-acid fast decolorized bacteria will take up that counter stain and appear blue at the end. So again, you're going to get one of two results, either pinkish, purple, so think fuchsia, or a blue color. And so pink is acid fast and blue would be non-acid fast. So the next picture is a slide, and this is a smear that we made in our lab that has both types of bacteria present. So there are both acid fast and non-acid fast that are mixed together on the same slide. And so hopefully you can see there is this pink color. So these bacteria in this area are that fuchsia color. So those would be acid fast bacteria. But then you have clusters of blue. So here's a blue, and then here's some blue and fuchsia intermingle. But you could then point to any bacteria and say, oh, that bacteria would be non-acid fast because it's blue, whereas this bacteria would be acid fast because it's fuchsia, or that magenta color. So that's our second differential stain. And then our third is called an endospore stain. So this too is a differential stain. So there's going to be two different dyes used, and at the end result, you're going to get two different colors. And so this is not differentiating between bacteria per se, but we're looking for the presence of an internal structure. So this is a genus, it's called Bacillus, and this is a different genus called Clostridium. And these are the two important genera that we know that can make these structures that are called endospores. And so what endospores are the word endo tells you something. So they're internal, so they're these structures that form inside of each of these genus of bacteria, and they allow the bacteria to survive really harsh environmental conditions. And so Bacillus and Clostridium can form these endospores. They're not for reproduction. It's just a survival mechanism that they have evolved. And the other reason or an additional reason endospore stain is important, is these two genera, Bacillus and Clostridium, are known to cause disease. So the disease anthrax is caused by a bacterium within this genus, and there's a variety of diseases, including botulism and tetanus, that are caused by this bacteria. So knowing if there's an endospore producing bacterium present could be helpful in identifying these two disease-causing genera. So endospores, by definition, are very resistant to things, and so they are also hard to stain, very similar to what we saw for the acid fast. So endospore stain is going to require the force, requiring us to force the primary dye into it. So that's what we see first. The primary stain is called malachite green. If you haven't noticed, many of these dyes have their color integrated into their name, which is helpful. So this is a green dye. And this will fairly readily stain bacteria green, but it won't stain the endospore unless we come up with a way to force it in. So same method as before, a long exposure time, so 5 minutes plus another 5 minutes, so it's 10 minutes in total. And during the first 5 minutes, we have a heat being applied. So we're really trying to force that first dye into that endospore. So if you look at this schematic, these are two different bacteria, what they're supposed to represent. This one has an endospore inside of it. That's what that circle means. Whereas this one doesn't have an endospore. So both of those bacteria will take up the green dye. And because we've forced it in using heat and length of exposure, the endospore also will have that dye embedded within it. There is no decolorizer here per se because malachite green is water soluble. And so when you rinse these bacteria with just water, this is deionized water is what that stands for, the green color is going to be washed out of the bacterial cell, but because that endospore is so impermeable to things, the water will not simply remove the malachite green from that endospore. So that endospore will still be green, but all the bacteria will be colorless at this point. So the second stain that's used, again, it's called the secondary stain or the counter stain. This is called safranin. So we saw this in our gram stain. It's a pinkish red stain. So that will be taken up by all the bacterium. And you're going to get several different possible outcomes. So you can see just red bacteria, and those would be bacteria without an endospore. Sometimes you'll see red bacteria with a little green speck in the middle, so that means the endospore is still inside. But over time, the bacteria that have an endospore, the cell part is going to fade away. And so what you're going to be getting is just this little endospore. I don't have my green pen handy, but it's just going to be a speck of green. So that's what this slide kind of shows us. There are some bacteria that don't have an endospore. So most of these are elongated bacteria. They're called broad in shape. Don't have green in them, so they haven't formed endospores. But there are some, like this one has a little sliver of green in it. So too does that one and that one as well. And then we have some where the green is just by itself. So these are endospores that are no longer inside of a bacterial cell, but are just now dormant in the environment. And just to give you some idea, these endospores are the most resistant bacterial structure that we know of. And so they can resist many disinfectants. So they're really hard to get rid of. They can survive years to hundreds of years just sitting basically dormant in the soil, and can undergo a process called germination to give rise to a living bacterial cell that can cause disease. So that's the endospore stain. So we have 3 examples of differential stains that we discussed: gram stain, acid fast stain, and now finally the endospore stain. There's one more stain that I want to introduce you to, and this is, here it's entitled a capsule stain because it's telling you what structure we're trying to stain within a bacterium. There's another name for this. It's called a negative stain. And the reason I bring that to your attention, because all the previous stains weren't classified as negative stains because, let's go back. The structures that we're interested in, like here the bacteria is red, here the spore is green, And so the structure that we are interested in has the dye color. A negative stain, one example is a capsule stain, the structure that you're interested in has the absence of color. So it's going to be clear where everything else that is going to be stained. And so in this case, a capsule is going to be an external structure that some bacteria have. So here's a picture where this purple is going to represent the actual bacterial cell, but then this clear kind of halo that encircles it is going to be a capsule. So not all bacteria have this, and so we can use this stain to ask the question, does this bacteria have a capsule, yes or no? And so you can put it through this stain where you apply this negative stain. Crystal violet is the color here. So we apply this purple color, and hopefully you can see, like, this one has got a pretty good example. There's a rod-shaped bacteria in the middle, and then there's a nice clear halo encircling it, which would indicate the presence of a capsule. And so you can see these littered throughout this field of view. So for the most part, we have purple over our entire field of view, but that area that lacks purple is the structure we're interested in. So this bacteria has a capsule. So that's an important, another important stain that we can use. I've been using these terms like round and rod, and so while those capture the shape, another word for shape is morphology, so I'll use those interchangeably, so you'll want to know both. That's one of the really important characteristics of a bacterial. So if we're trying to identify a bacterium, we want to be able to make a smear of it and stain it and then view its shape and size. And so the two most common names that we're going to use is if something looks pretty much spherical, we're going to call it cocci in shape, or you can use the word coccus. So the only difference between these, obviously they're spelled differently, but they mean the same thing, roughly spherical. And it's just singular versus plural. So it's like octopus and octopi. So cocci would be more than one. So if you had a cluster of bacteria that are roughly round, you'd say, oh, look at all those bacteria, they're cocci in shape. If you had a single bacterium, you'd say, oh, that bacterium is coccus in shape. If you don't use those perfectly, if you misuse them, singular versus plural, I'm not going to hold it against you, but that's the difference. There's the other common shape is bacillus, which is rod, and same sort of singular versus plural. One bacterium that's rod shaped would be a bacillus. And then if there's a bunch of them, we'd say, oh, those are all bacilli in shape. So bacillus, singular, bacilli, plural. This is a term that's used by some microbiologists. I'm not going to really use it, but just realize it exists. If something is round, it's cocci. If it's rod, if it's elongated to any extent, many people will call that bacilli. But some use this intermediate. So if it's slightly elongated, it's coccobacillus. But anyways, so that term does exist. So by far, cocci and bacilli, those are supposed to be stars that I'm making there, are the two that we see most frequently. But realize there are some other shapes as well. So Vibrio is going to be a rod, but it's slightly curved. So that's that shape. These two are similar. They're both going to be spirals or corkscrew in shape. So this 2D image doesn't really do it justice, but you have to imagine that's got kind of a corkscrew to it. The difference between them is the relative thickness of them. So spirillum is thicker in width, so a thicker, wider corkscrew, whereas a spirochete is going to be thinner. And so that imparts different physical characteristics in terms of their structure. So thicker means a stiffer spiral, thinner means more flexible. And then the last shape is not really a shape per se, but we use it to describe bacteria that don't have a characteristic size or shape. So for example, we can say, oh, for example, strep infections, they're always going to be round in shape. So we'd say, oh, they're cocci in shape. Clostridium, so botulism is always going to be bacilli in shaped. But there are some bacteria that don't have a characteristic size or shape, and so to describe that, it just means many shapes. So pleomorphic means multiple shapes. So if you see that term to describe a bacterial species, it means it doesn't have one shape to describe it. Shape or morphology of a bacteria is really important. So that coupled with a gram stain are kind of the primary things that we want to look at for a bacteria and identifying it. But there's one other thing that we also look at, and it's related to how bacteria reproduce. And so this slide gives us a quick introduction to how bacteria are going to divide. So all bacteria are going to use asexual reproduction, so they do not reproduce sexually. And so there are multiple ways that they carry out asexual reproduction, but by far the most common is binary fission. So binary fission is going to involve the steps that are seen over on the right. So what you start with is a single bacterium, and what's going to happen is their genetic material, their DNA, their instruction manual has to be copied. And so that's what we see here, the replicated DNA. And that has to happen prior to this binary fission or cell division. The bacterial cell gets a little bit bigger, but not super big. And then that just allows enough material that it can be divided into two. So you have this cross wall that starts to form until it totally separates the two cells. And each of them, we call these daughter cells, have the complete instruction manual in it. So this is the bacterial DNA. So this is what we're interested in, kind of this step. So some bacteria separate, and so these individual cells are free of one another, but sometimes bacterial species will remain attached together or they fail to separate. And if that happens, then we can generate what are called bacterial arrangements. So arrangements occur during binary fission if those daughter cells fail to separate. So for example, if you have this one bacteria that is cocci in shape, so that's round, and if it divides along this plane, in this case, I have it drawn vertically, what you're going to get is one bacteria on either side. And if they fail to separate, we now have what's called an arrangement. And so arrangements are determined by two things during binary fission. One is if the bacteria fail to separate, you generate arrangements. And then also how the planes of division occur. So what I mean by that is if another round division occurs, if the planes go in this direction again, whoops, sorry about that. If they go vertically again, then you get a specific arrangement that we'll see on the next slide. Or if you get division happening on this more horizontal, then you get a completely different arrangement. So let's look at those arrangements that are applied to cocci cells. So here is an example of an arrangement. It's called Diplococcus. And this is when you have pairs. And so you just have pairs of cocci. And so this is the actual stain. So here's a pair, there's a pair, and there's a pair. So it's called Diplococcus. And so that means that when they divide, pairs tend to be stuck together. And so during division, failure to separate, you can get Diplococcus. Streptococcus is when you get division along the same vertical plane over and over again, but you get a chain. So this is a chain of cocci. That's what Streptococcus means. And so here we have, that's a single bacteria, then it's a second bacterium, and a third. And these are organized end to end, and so they form this chain. So that's what Streptococcus means. Then we have tetrads, which is kind of a cluster of 4. So there's a tetrad. And then kind of a cube is sarcina or sarcina, depending on how you pronounce it. These names, while they do exist, we don't see them too often. Okay, so tetrad and sarcina, not the most important, but Diplococcus, Streptococcus, and Staphylococcus are the 3 most important arrangements that we're going to see throughout the semester. So those are going to be the most important. And so I haven't explained what Staphylococcus is. So this is individual cells are round in shape. That's the coccus part, or cocci. But these are known as grape-like clusters or irregular clusters. So one difference that we can say exists between bacteria that cause strep infections, So strep is short for streptococcus. And staph infections caused by staphylococcus, one difference between them is how they look under the microscope. They have the same shape, both round, both cocci. So that's in their name, coccus. But staphylococcus is going to tell you these are grape-like clusters, whereas streptococcus are going to be organized into chains. So these arrangements describe bacteria that are cocci in shape, but we can use the same terminology for bacillus. So rod-shaped is a bacillus. So here's the micrograph. So picture through the microscope, and you can see these are elongated like little dashes. So that's a single bacillus. They don't look like they're attached to one another. Here is an example of diplobacilli, and so that's again an example where we have pairs, and so these are end-to-end on the chain. I'm sorry, on the rod. So diplobacillus is you have two bacilli end-to-end. Strepto is chains again, but notice now we're calling this streptobacilli because you have chains of rods. So streptobacillus. And then you can also have bacteria stacking on kind of this axis, so kind of like how pancakes would stack. That's called a palisade arrangement. And then also you can have kind of ends attaching, so they're not, we would call this diplo, but instead we call it a V shape. So diplo, they're kind of nice in a row, whereas V-shaped is going to be angled a little bit more. So I split this one into two just to make it a little bit more manageable. In addition, there were some technical difficulties, so I had to redo the second half. So moving through this lecture is really understanding how we can name and identify bacteria. And so part of that is using microscopy to visualize bacteria. And so to be able to see bacteria under the microscope, we have to make bacterial smears, which is putting them onto a slide, and then stain them either using a simple stain, or often we use a differential stain like a Gram stain. And so at the end of the staining procedure, you get multiple pieces of information for a given bacterium. So you get the size of the bacterium, but you also now can use terms like the shape or morphology. So you can say, oh, this bacteria is bacillus in shape, or it's vibrio in shape, or it's coccus in shape, or whatever the shape indicates you to conclude. But then above and beyond the shape or morphology of a single bacterial cell, you can also ask the question, is there an arrangement? So are they organized in chains? So that coccus is in chains, so it would have an arrangement of streptococcus, or it's in clusters, it's staphylococcus, etc. So that's one really important piece of the puzzle to understand and identify bacteria. So next lecture, we're going to talk about the prokaryotic cell structure. And so that's what we're going to do.